You know, the human heart, it's an absolute masterpiece of biological engineering. You've got four chambers, four valves, all working in perfect sync from before we're even born. But what happens when that original blueprint, the one for this incredible little machine, has a typo? A single tiny error that can change absolutely everything. So today, we're going to dive into the hidden genetics behind these typos and figure out what they really mean for the heart.
And get this, it happens way more often than you'd probably guess. About 1 in every 100 babies is born with what's called a congenital heart defect, or CHD. That makes it the single most common type of birth defect out there. It's a staggering statistic, really, and it's one that has kept doctors and scientists searching for answers for decades. Now, when we talk about congenital heart diseases, it's not just one thing. We're talking about a whole spectrum of structural problems. They can range from tiny little holes that might even close up on their own, all the way to incredibly complex malformations that demand immediate, life-or-death surgery the moment a baby is born. Let's just take a look at two of the most severe forms to get an idea. There's Hypoplastic Left Heart Syndrome, where basically the entire left side of the heart just fails to develop properly. And then there's Tetralogy of Fallot, which is actually a combination of 4 different defects all at once. Both of these conditions completely wreck the heart's carefully controlled circuit, and it wasn't that long ago that these were almost always fatal. Okay, so let's break down what's actually going on in Tetralogy of Fallot. In a normal heart, you've got this solid wall separating the two main pumping chambers, keeping oxygen-rich and oxygen-poor blood apart. But in TOF, boom, there's a big hole in that wall. On top of that, the aorta, the main artery that should be sending all that good oxygenated blood to the body, is shifted over and sits right on top of this hole. Add in a narrowed pulmonary valve, and the result is that oxygen-poor blue blood gets pumped out to the body, and that is what leads to the classic blue baby syndrome you may have heard of. For years, you'd think the answer was simple, right? Just find the one broken, bad gene that's causing all the trouble. But the genetic puzzle of CHD has turned out to be way more complicated than that. It's not about one single error anymore. It's about a much more intricate network of instructions that have all gone wrong.
And here is the biggest clue, the thing that really changed the game. Genome-wide studies have uncovered something absolutely fascinating. More than 90% of the genetic variations linked to cardiovascular diseases, they aren't even in the genes that code for proteins. Nope, they're located in these vast, non-coding regions of our DNA. Yeah, this is the part of our genome that scientists for the longest time literally called junk DNA, but we now know it is anything but junk. Here's a good way to think about it. Imagine your genome is a giant cookbook. The protein-coding genes, those are the recipes, the instructions for making things like muscle or enzymes. But the non-coding genome, that's where the crucial instructions are. It tells you how hot to set the oven, for how long, and for which meal. It's the master regulator of the whole kitchen.
So what happens when there's a typo in those instructions? Let's dive into a specific case study to see this in action. Researchers wanted to know how one tiny little change in this non-coding DNA could have such massive consequences for a developing heart.
So how did they do it? Well, first they used these incredibly powerful computer models to scan the non-coding genome, looking for thousands of tiny single-letter changes, what we call SNPs, that were linked to heart disease. Then, they used those models to predict which of these typos were most likely to mess up the landing pad for a really critical heart development protein called GATA4. And finally, they took those predictions into the lab and confirmed it. The variants completely blocked GATA4 from being able to latch onto the DNA.
It really is like a broken light switch. The GATA4 protein is like your finger, trying to flip a switch to turn on a gene that's essential for building a healthy heart. But that single-letter typo, that SNP, has completely changed the shape of the switch. Your finger can't connect, the switch never gets flipped, and that crucial gene, well, it just stays off, in the dark.
Okay, so a broken DNA switch, that's one major piece of the puzzle. But what if the problem isn't a typo in the instructions at all? What if a totally different player in our genetic story just goes missing? What if the conductor of the entire orchestra just vanishes? We are talking about something called long non-coding RNAs, or LNC RNAs for short. Now, these molecules don't make proteins. Instead, they act like signals and scaffolds, organizing huge networks of genes. The research we're looking at focused on one in particular, GATA6 AS1, and you can absolutely think of it as the conductor of a genetic orchestra that's supposed to be playing the symphony of heart development. So the obvious question is, what on earth happens to the orchestra if the conductor suddenly disappears? What kind of music does it end up playing then? Well, in mouse studies, the answer was pretty dramatic. When scientists removed this LNC RNA conductor, the developing heart completely failed to form a normal 3-leaflet aortic valve. Instead, it formed a 2-leaflet valve, which just happens to be the single most common congenital valve defect in humans. The missing conductor threw the whole genetic symphony into chaos, and the result was a major structural problem.
So these two case studies, the broken switch and the missing conductor, they're really just two pieces of a much, much larger and more intricate puzzle. What they show us is that the blueprint for the heart is written in a language that is far more complex than we ever imagined. This really shows us that there isn't one single cause of CHD. It is a multifaceted, polygenic problem. As you can see here, you might have a single-letter SNP that acts like a broken switch. Or you could have a missing link RNA conductor. Or you can even have something called a copy number variation, where entire sections of DNA are deleted or duplicated, which completely changes the dosage of critical genes. It's coming from all different angles. And that really brings us to the key takeaway from the research. As the scientists put it, the most plausible explanation is an accumulation of these alterations. In other words, it's not about finding one big broken gene anymore. It's about understanding a complex system that's been pushed out of balance by lots of small changes all across the genome. And this all leads us to a final and pretty provocative thought. For decades, we searched for answers in just 2% of our DNA, the part that makes proteins. But what if the future of understanding and maybe, just maybe, one day treating these devastating diseases lies in decoding the complex regulatory language that's been hidden all along in the other 98%? The very part of our genetic blueprint we once just dismissed as junk?